Hello everyone, my name is Samantha Sherman. I am here to present my research on optimization of in vitro biomass production of the anxiolytic herb Scutellaria lateriflora, commonly known as American skullcap. This plant has anti-inflammatory, antispasmodic, and anti-tumor properties. It is traditionally used to treat insomnia, anxiety, and nervous tension, and found in the form of tea, extracts, tinctures, and capsules. The demand for skullcap products is rising. Organic American skullcap costs $120 a kilogram. Toscan et al. In 2010 compared the growth of Scutellaria lateriflora along with two other Scutellaria species using different culture systems and showed the importance of liquid cultures and the use of fiber support for high-quality biomass production devoid of hyperhydricity. We tested 2 carbon sources at 4 concentrations and the effects of 4 cytokinins in shoot induction and biomass production using 3 culture systems. The role of carbon source was tested using MS media supplemented with sucrose or maltose at 0, 3, 5, 7, and 10% concentrations in semi-solid, liquid stationary with fiber support, and liquid agitated with fiber support culture systems. Liquid agitated with fiber support system using 3% sucrose was the best system for biomass production and in vitro plant quality. After 6 weeks, explants grown in liquid agitated culture had approximately double the biomass in comparison with semi-solid medium. Liquid agitated cultures produced 405 milligrams of dry weight biomass compared to their counterparts grown in semi-solid medium, which produced 206 milligrams of dry weight biomass. Liquid stationary system had the lowest biomass recovery, and this result followed Toscan et al.'s work in 2010. Once carbon source increased to 5% or more, hyperhydric plants were evident, which increased with sugar concentration, and this can be seen in Figure 3. The role of cytokinins on biomass was studied using semi-solid MS medium with 3% sucrose supplemented with 4 cytokinins, kinetin, BAP, metatoplin, and TDZ at 0.1, 1, and 10 micromolar concentrations. Biomass experiments were carried out using 20 explants per treatment and 4 replicates for each treatment. Explants were weighed after 6 weeks in bundles of 5 explants per bundle, and the same amount of media was used in proportion to the number of explants per culture vessel in all biomass experiments. In semi-solid medium, 10 micromolar BAP was the best for biomass recovery, producing 345 milligrams of dry weight biomass. The control MS medium without cytokinins produced 166 milligrams of dry weight biomass. Next, 10 micromolar BAP and 10 micromolar metatoplin supplemented liquid agitated media were tested for multiple shoot induction and biomass production. MS liquid agitated system supplemented with 3% sucrose without cytokinins was the best treatment. We also studied the role of 3 cytokinins in semi-solid MS media for shoot production using BAP, metatoplin, and TDZ at 0.1, 1, and 10 micromolar concentrations. Shoot induction was highest in MS medium supplemented with 10 micromolar metatoplin, producing an average of 4.5 shoots per explant and more than double the shoot count of the control, which only produced 1.9 shoots per explant. Acclimatization was carried out using rooted explants, which were transferred to high humidity magenta boxes filled with sterile potting mix and later transferred to the greenhouse. And plants had an 87% survival rate after being transferred to the greenhouse. Previous studies have shown the importance of using fiber support in liquid culture systems. Our study has gone further by investigating 2 different carbon sources at different concentrations, as well as the effects of 4 different cytokinins using these culture systems. A liquid agitated with fiber support was best for biomass and plant quality compared to other systems that were tested. Liquid systems provide greater availability of water, nutrients, and plant growth regulators as a result of lower resistance to diffusion and closer contact between explants and medium. We also found that sucrose is a more efficient carbon source compared to maltose. Sucrose is a non-reducing sugar, which breaks down into 1 molecule of fructose and 1 molecule of glucose. Maltose breaks down into 2 molecules of glucose. Glucose is used in many other metabolic processes, but fructose can be used specifically by the plant to increase biomass. In conclusion, an efficient micropropagation and acclimatization protocol was developed in this study for increased biomass production of high-quality in vitro micropropagated Scutellaria lateriflora without the use of agar and plant growth regulators, which can greatly reduce costs for large-scale production of this medicinal plant. Thank you very much.